Uh, Good morning, everyone. I'm here to present some of our experiences on the ecology of macrofungi in Southwest India. It is, although it is of ecological aspect, it is more of our experience for the last 1.5 years, 1.5 decades, working on different aspects of macrofungi. And I'm thankful to my students who has materialized my ideas and also to put into publications and so on and so forth. So we all know about macrofungal organisms, and they are highly fascinating, and they are very useful natural resource as food, and also health benefits, and also they are agriculturally important and industrially important. And currently, approximately 2.2 to 3.8 million is the fungal population, but the macrofungal population ranges between 0.14 to 1.25 million. Although we have so much of fungal population, what we currently know is less than 10%, and this less than 10% that has been explored, and we have got this much of benefits from these organisms, and you can imagine how much potential it has with regard to further exploration of these macrofungi. This macrofungi has a lot of ecosystem services, as everyone is aware of that. First and foremost is nutrition, and they produce a lot of metabolites, and mainly they are decomposing organic matter and mineralize this organic matter, and they involve in biogeochemical cycles, and they associate with plant species as ectomycorrhizae, and provide nutrition to the plants, and they protect the plant, and plant growth promotion is possible, and they are responsible for stress tolerance of plants, and they are also very useful in soil stability and bioremediation, and also they enhance the carrying capacity of the of a specific ecosystem where they exist. So this information will give you an idea that how these macrofungi are dependent on environmental variables and also substrates and in association with life forms. So there are so many environmental variables. They are controlling the growth and perpetuation of macrofungi. For example, temperature, season, rainfall, soil fauna, so on and so forth. Then with regard to substrate, there are plenty of substrates on which macrofungi can grow and propagate themselves on leaf litter, logs, fruits, insects, termite nests, and so on and so forth. In addition, macrofungi also associate with several life forms. For example, plants, as I mentioned earlier, they are mutualistic and they become ectomycorrhizal and useful one. And nevertheless, some of them are parasitic too. And with regard to animals, they are mutualist. With best example is ectosymbiosis with termites, and some of these macrofungi are also parasitic on animals too. With regard to the substrate, although we classify substrate into different categories, under each category there are so many subcategories, and they are important to understand the importance of growth of macrofungi. If you take the example of soil, you can think of the anthills, earthworm casts, termite mounds, sandy soil, loamy soil, like that. In wood, you can classify into different categories: fine, medium, and coarse woody materials. And that is the woody material is perpetuating, and it is stable and support several macrofungi, and it is a sort of many of them grow seasonally on wood material. And leaf material is very interesting material where we can see petiole, midrib, and lamina, and the leaf litter. Leaf material is very important for Marasmius and Marasmiellus species can be evaluated very clearly. And dung, with regard to ruminant dung and also non-ruminant and insect excreta, substrate for macrofungi. Several adult larvae and dead part of insects also supports the macrofungi, and many of them prefer different substrates. They may prefer single substrate, and they may prefer two substrates or multiple substrates. For example, our experience is that with regard to coastal sand dunes, sandy soil supports nearly 30, more than 35 species. Wood supports approximately 20 species, 20 and about 20 species. Leaf litter is comparatively less, and some of them grow on soil as well as leaf litter. Some of them grow on leaf litter as well as twig. Some of them conquer soil as well as wood. Like that, sometimes it is dual and multiple substrates be preferred by many of the macrofungi. If you look into the ecosystems of Southwest India, everybody is aware of that. A lot of forests are there, plantations are there, forest reserves are there, shola forests. Shola forests are very interesting because they are in the valleys of mountains. They cover the valleys of mountains, and that is referred as a special forest, referred as shola forest. Sacred groves are they have been preserved for various purposes. Coffee agroforests are plenty. If you look into coastal plains, we have scrub jungles and also plantations, and even grasslands also there. And some of the institutions have established arboretum, housing some of the endangered and endemic plant species in arboretum. Many plantations, agroforests, and also swamps also exist in coastal plains. In mangroves, as everyone aware, estuaries are proper estuaries, and some of the islets can be seen, and bays, lagoons, and a lot of mangrove tree species and mangrove associates also there in the mangrove ecosystem. These are all provide substantial material for growth of macrofungi and other fungi. If you look into coastal sand dunes as such, and natural vegetation is plenty, and commercial plantations also there that adjacent to coastal sand dunes. And some of these macrofungi prefers two ecosystems or multiple ecosystems. So therefore, it is interesting to understand how these organisms have adapted to different ecological conditions. So with regard to the statistics, whatever available in the literature, varieties of diversity and phylogeny and distribution has been reported by many of them, especially from the southwest region. That is, Ranadive has reported nearly 175 species of Aphelophorales in 190 genera, and polypores are plenty in Kerala, 135 species. Agarics have been reported in Maharashtra and Kerala. Non-gilled fleshy fungi have been reported in Kerala, and Mycena has been reported nearly 50 species in 15 sections. And Manimohan and his group published a book on Mycena. It is available. Anybody can procure it from the research gate. Then with regard to ectomycorrhizas, Western Ghats has nearly 150 species. Kerala, they have reported nearly 30 species. Sahib, again, Manimohan and his group has published a book in Spore Print publishers. It is available in the research gate. So this is all these aspects pertain to more of taxonomy, physiology, phylogeny, and checklist rather than the ecological studies. So therefore, I thought that it is better I can project some aspects pertain to ecology. So this is we have from last more than a decade, we have evaluated 8 different ecosystems in Western Ghats and West Coast and mangroves. So the reserve forests, shola forest, sacred groves, scrub jungles, arboretum, botanical gardens, mangroves, and coastal sand dunes. So here, the this picture indicates the total number of species known, and also it is a known species and also unknown species. Unknown refers to they have been identified up to genus level, not up to species level. Known species are approximately 30 here. And if you look into coastal sand dunes, the number is very close to 40. And unknown also high, maybe around 25 species are unknown based on the report. And here you can show the forest shows maximum of 85.4% identified species. Sacred groves comes next to Shola forest. Arboretum 76.7%. Reserve forest 70.7%. Rest of the ecosystems are less than 70%. And here the unidentified species, coastal sand dunes is 40.6%. Then botanical garden 38.9%, scrub jungle 35.3%, mangroves 32.6%. So this unknown species main because it shows that the capacity of the ecosystem to project various species, macrofungal species, they are in need of further verification and also identification and bring into mainstream of advantages. I would like to project here. This is the number of species. This is the sporocarp per quadrat, 25 into 25 meter quadrats. So the number of species is not matching with the sporocarp number. For example, this is the lowest number of species, total species observed in Arboretum, but Arboretum has maximum number of sporocarps, more than 400, 25 into 25 meter quadrat. And similarly, here it is a high species, that is Shola forest, and it has close to 400 quadrats per quadrat. And here, interesting thing is coastal sand dunes has nearly more than 60 species, but the number of sporocarps per quadrat is less. It is approximately less than 75 per quadrat. So therefore, the number of species is definitely not matched with the number of sporocarps in a defined area of survey. Yeah, this is seasonal species and sporocarp richness has been projected here. From that is in the wet season from the month of June to November. Of course, Western Ghats receives monsoon, southwest monsoon in the month of May itself, maybe mid or end of May or early May, or sometimes we may get pre-monsoon shower in April itself. So the reserve forest, Shola forest and sacred grove shows almost a similar pattern with regard to seasonal changes. There is no much difference in this. But if you look into these scrub jungles, arboretum, and botanical garden in the coastal region, the botanical garden and scrub jungle shows similar pattern with minor changes. So there is a spell here, that is July, it is more number of species, and again there is another peak, small peak somewhere in September. But the arboretum is something, it is an outlier. So it is not going to follow the pattern what we have seen in case of scrub jungle and botanical garden. It has two important spells, that is in the month of June and in the month of August. And the total number of species are relatively very high compared to scrub jungle as well as botanical garden. Most likely it is because a lot of endemic and endangered species have been housed in arboretum, and that may support varieties of macrofungi. And if you look into the species richness and in mangroves and coastal sand dunes, so they show highest peak in the month of June, where rain is very high, and there is a small spell here, and again it decreases, or in case of mangroves it increases. But if you look into the number of species in Western Ghats, it is 30 plus, 20 to 30 species. And whereas here it is 10 to 20 species, 20 species in Arboretum, whereas here it is 10 species approximately. It comes down, whereas here close to 20, and here it is 15 species in case of coastal sand dunes and mangroves. And here I have projected here the sporocarp richness. Arboretum, as I mentioned, it is very high number, Shola, followed by Shola forest, reserve forest, sacred grove, and rest 6 ecosystems less than 100 sporocarp per quadrant. Species richness is not matching with this sporocarp richness, and Arboretum is an outlier. Probably this may be interesting point to note that such type of arboretum are there in many locations in India, and also where they deliberately they have incorporated a lot of endangered endemic species that is going to support very high number of macrofungi. And this is the Venn diagram shows that this is actually not rainforest. This is the reserve forest and Shola forest and sacred grove. When they have been compared, the number of overlapping species are very less. One species here, that is between rainforest, the reserve forest and Shola, whereas in case of sacred grove, is two species, whereas Shola and sacred grove only 3 species, and no species common to all the 3 locations, 3 ecosystems. If you come to these coastal plains, I have compared scrub jungle with arboretum and also botanical garden. The number is slightly higher than Western Ghats. The overlapping species, 6 species here, 13 species between scrub jungle and botanical garden, arboretum and botanical garden 5 species, and 2 species were common to all the 3 ecosystems. And this indicates that there is something special with regard to Western Ghats are concerned, because these ecosystems are independent and they have their own potential with regard to processing the macrofungi compared to that of coastal sand dunes. But coastal sand dunes have some very important macrofungi too. And if you look into maritime habitats, mangroves and coastal sand dunes, 21 species are overlapping. That shows that there is some similarity with regard to mangroves and coastal sand dunes, but salinity may be one of the factors. Of course, mangroves are getting fresh water too. By that, depending on the fresh water, macrofungi may grow, and some of the tolerant species also grow like that. And here, if you look into the core group species, core group fungi refers to those they are occurring more than 10% of the total species, total number, that is frequency of occurrence, more than 10%, and no or less overlap of core group species. So maybe 1 or 2 examples are there, but not many. But we can take it as no much overlap of core group species in these 3 ecosystems. With regard to core group species, Shola shows the highest number of core group species, that is 11 species. Reserve forest followed by reserve forest, arboretum, botanical gardens, sacred grove, mangroves. Coastal sand dunes has only 1 species, Dacaryopinax. It is very prominent in the west coast. And scrub jungle, where we can Xylaria hypoxylon is very common. And in fact, the scrub jungle of west coast is a hub of varieties of Xylaria species. We have come across more than 20-25 species of Xylaria, and only 1 core group species overlap between reserve forest and Shola, as I mentioned. That is the Microcarpus vernicis. Vernicis is the only one that is overlapping. Rest of the core group species never overlap. So that is a very interesting point to remember. With regard to entomopathogens, there are many entomopathogens that has been reported from the Western Ghats. 15 species of cordyceps have been reported from Maharashtra, Karnataka, and Kerala. And varieties of cocoons and larvae and insects have been attacked by entomopathogenic fungi. And in Karnataka and Kerala, you know very well that coconut is very important plantation crop, where there is a coconut grub is referred as Leucophilus species that is attacked by the cordyceps species. Recently, it has been reported in Karnataka as well as Kerala in the coconut plantations. In Karnataka and Maharashtra, there are pentatomid bugs, especially Heliomorpha halis, that is stink. It is referred as stink bug, and it has been attacked by Ophiocordyceps nutans. So everyone is aware that these cordyceps and allies are very, very important with regard to metabolites are concerned, and also control of entomopathogenic fungi, entomopathogens that they are detrimental to plants. This is the photograph shows that we have identified, and identified means not completely identified. We are trying to go for the molecular approaches to find out what are these cordyceps and allied species, and some of them grow on the grasshoppers and some larvae here. You can see that, and this fungus has grown on a millipede. A millipede, this is the millipede body, dead body, and the cordyceps has emerged out. Some of them cocoons, oval type of cocoons and spherical cocoons. And another insect may not be cordyceps, allied species is growing in subjungle. And from the last 5 years, we are monitoring different types of cordyceps in this area, and we have added nearly 10 to 15 different morphologically different cordyceps in the subjungles. And this is Ophiocordyceps nutans in the Western Ghats. They grow on this, I have mentioned this species, Heliomorpha halis, that is the insect stink bug. And these are the stink bugs. From the thorax region, it emerges out, and the sporocarp will be produced. And these are the sporocarps of Ophiocordyceps nutans. And so this has a sort of association. Tripartite association is with the plant species and also the insect species and the fungus. So this is Cassina glauca tree species, which is not an endangered or endemic species. It is common in Asia and also Southeast Asia. Even India, it is distributed, very commonly distributed. And this is the insect, the dorsal view of the stink bug and the ventral view of stink bug. And this is the plant leaves and inflorescence and the fruit. And when you cut the bark of Cassina glauca, it gives a sort of a red colored pigment bark. And these insects are going to damage the bark because they are sap-sucking nature. And it is going to damage the bark, and it is rejuvenated. But sometimes we can see highly damaged part of the bark because of the stink bug. And in fact, this Cassina glauca bark consists of the Ophiocordyceps nutans anamorph stage, that is hirsutella. And because these stink bugs are sucking, and they are invariably get this anamorph into their gut, and afterwards in the gut they are going to establish the perfect state is going to establish, and from the thorax region, the Ophiocordyceps is going to emerge. On many occasions from the abdomen region and even head region also, but by and large it will be from the thorax region. The Ophiocordyceps emerges out. Let us look into the coprophilous fungi. With regard to coprophilous fungi, not much work has been carried out in the western part. The real elephant tongue is major coprophilous fungi reported from Karnataka as well as Kerala. Up to 35 species have been reported. And in the elephant dung, there are several macrofungi. They are hallucinogenic in nature, especially Conocybe and such type of macrofungi, and they are psychedelic in nature. And the major species or the Paniolus, and also Conocybe, and also Strophora. These are the three species very common, and rest of them, one or two species have been found. In Kerala, cattle dung has supported three species. This is very low because not much of work has been carried out on macrofungi in the western part of India. That shows that it is one of the virgin areas where people can take up such type of studies, especially with regard to macrofungi. And this is the picture shows different types of um, macrofungi growing on elephant dung. You can see that Conocybe, Coprinus, this is Coprinus, and Pseudosardaria, is an Ascomycetes fungus, and also psilocybe. Psilocybin is very important bioactive compound in psilocybe. Like that, there are various species have been reported from the elephant tongue. With regard to ectomycorrhizas in the Western Ghats, so the global estimate of ectomycorrhizae, approximately 20 to 25,000 species are there. But we according to our survey, we got nearly approximately 150 species of 34 genera, and they colonized nearly 60 host tree species belonging to 40 genera, and consisting of major ectomycorrhizal fungi include Elates, Amanita, Russula, and also Inocybe. Um, we have nearly, it is interesting to note that among the 148 species of ectomycorrhizae, 114 species, it is approximately 77% is associated with the Dipterocarpaceae members. So Dipterocarpaceae members are very, very important with regard to supporting the ectomycorrhizal fungi. And that shows that Dipterocarpaceae is very well known, especially for the presence of ectomycorrhizal fungi. And Veteria indica is one of the important Dipterocarpaceae members, showing up to 69 species of ectomycorrhizal fungi. Of course, there are Hopia ponga, Hopia parviflora, Myristica, Malabarica, Dipterocarpus indicus. These are all Dipterocarpaceae members. Nearly there are some 78 species of Dipterocarpaceae members are present in Western Ghats, and they support a variety of macrofungi. Therefore, it is very important to conserve such type of plant species. And the benefits of this ectomycorrhizae, some of them are ectomycorrhizal, and many of them are edible, and also many of them are edible. Therefore, it has medicinal. Therefore, it is of special interest to look into multiple benefits of ectomycorrhizal fungi. So this this information shows that this is general feature of 148 species of ectomycorrhizae in the Western Ghats, that is the tree species. So the first major species Inocybe, and then Russula, and third one is the others, one species each, that is up to 17. Next one is Boletes and Lacaria, Sulius, Scleroderma, like that. It goes 148 species. So if you compile 114 species observed in Dipterocarpaceae, Inocypes are highest, like that of general pattern, 36 species, 30 species of Inocype, Russula 31 species, Dipterocarpaceae, and others 12 species. So the pattern is more or less similar with Dipterocarpaceae member. That is among 148 species, nearly 70, that is 114 species, comes to 77% is in association with the Dipterocarpaceae members. So this may be very useful information to many of us with regard to importance of Dipterocarpaceae in different ecosystems. This is the picture of selected ectomycorrhizae, mycorrhizas in the Western Ghats. You know very well this is the Astreus hygrometricus, mature species, and Astreus odoratus, and this is Boletellus species and Scleroderma species, and this is again Phlebopus marginatus, Phlebopus marginatus, and there are two species in Phlebopus. This is another Phlebopus. Erva lady is holding this. This is a very big mushroom. and this is Lycopterodon, and this is again Rosula, and this is Scleroderma and Telephora. So these are some of the species we have projected. That is the common ones are present in the as selected mycorrhiza. Scleroderma is of special interest in ectomycorrhizae because it is a cosmopolitan species, and nearly 8 species have been reported from Southwest India. The interesting point is they are ectomycorrhizal as usual and edible, and many of them are medicinal. And another property associated with Scleroderma is they are extremophilic organisms. So we can find them in the polluted habitats too, and some of the extreme habitats. So it has a broad host range, nearly 43 species, including exotic species. Exotic species including this acacia, then eucalyptus, and even many of these silver oak, and even casuarina. These are all some of the exotic species found in the even in Western Ghats as well as West Coast, where these species are associated with them, and they have adapted to xeric habitats. So where nutrients are less or this temperature is high, coastal sand dunes occupied by them, mining area has been occupied, especially coal waste dump, also sites for presence of Scleroderma. So therefore, Scleroderma may be, as they are ectomycorrhizal, they may be very useful in rehabilitating some of these threatened or human impacted areas. So these are some of the examples of Scleroderma, that is Scleroderma aerolatum. These two are Scleroderma aerolatum and Scleroderma cepa, the bovista, and this is Scleroderma polyrhizum, and this is Scleroderma duciosporum, Scleroderma citrinum. So like that, there are several species under Scleroderma, very common in the Western Ghats as well as West Coast. So let us look into termitomycetes. So termitomycetes are very useful and highly delicious macrofungi. Nearly 20 species are known from the Western Ghats, especially from Goa, Karnataka, and Kerala. And major species include Termitomyces clypeatus, eruseus, Termitomyces hemi, and Termitomyces microcarpus, or wide distribution. And many termitomyces are association with the Scleroderma and Xylem. It is interesting. See, these termite mounds are also occupied by Scleroderma and also occupied by Xylem. We can find termitomyces associate with in the termite mounds with Xyleria, and also termitomyces. Many species of termitomyces are found with Scleroderma, as we see in some of the Eastern Ghats as well as West Coast. These are some of the examples of termitomyces. That is clypeatus, because it is pointed umbo. Umbo is one of the important features to identify this morphological identification of termitomyces. And eruseus, and this is fuliginosus, and this is globulus, and this is hemi, and this is indicus. So these are some of these found in Western Ghats as well as West Coast region. And these are some additional species. Mammiformis. We found Mammiformis recently in our campus, Mangalore University campus. And this is also a very small one, microcarpus, but it is very delicious. And Termitomyces schimferi is very common in some of the coastal region. So the termite mounds of coastal region, several termite mount shows this Termitomyces schimferi, and that is one of the food material for many of the coastal people. And this is Termitomyces striatus, and this is Termitomyces titanicus. Termitomyces titanicus is very big. It diameter may be around 35 to 40 centimeter diameter. So this is the hand of a person who has hold this mushroom. You can imagine the size of the Termitomyces titanicus. And this is one, very common in especially in the coastal, our campus and the coastal region also we can find many of them. The interesting feature here is the life cycle of termites and termitomycetes are go hand in hand. So this is the sexual phase. Actually, this is the asexual phase where the nuptial flight of termites, the reproductive pair, is going to attack the termite termite mounds, and that will take the termite spores, and it will disseminate the spores, and spores will accumulate on the leaves and woody debris, and it is mixing with the fecal pellet, and the mycelia will grow. And actually, it is not a sexual phase; it is asexual phase. And once it is taken up further, the mycelia further will be utilized by the uh, inoculated in the comb of termite comb by the workers of foraging workers, and spore germinates and homokaryotic hyphae will develop and leads to fusion of hyphae by plasmogamy and karyogamy, and then termite fruit body, that is termitomyces fruit bodies are going to develop. So this is this life cycle of termites and the termitomyces is embedded here. So both of them are ectosymbiotically associated, and their life cycle is dependent on each other. So these are some of the pictures of the macrofungi found in scrub jungles. You can see the Amaroderma here. Ganoderma is very common. Geostrum is also here. And this is again, you can see Lycopterodon, and Ramaria is here, and Tramella. There are so many species exist in. This is again Xylaria, West Coast region. As I mentioned, Xylaria is plenty in the West Coast region. And this is the Xylaria complex, as I mentioned, Xylaria steroidea. It is very interesting. The tip is curved initially. That shows that it's possible to identify them. Xylaria hypoxylon, and then Xylaria filiformis is it has a small button-like structure at the tip, and that's how we can be identified. And Xylaria longives also very common. This is all digested longives. This is the young stage, and the multiplex is also there. So these are all common in the scrub jungles. And very recently, one of my students corrected a species known as Xylaria Sridhar. Actually, the one of my colleague is Dr. Sharat Chandra. He's um, he's working. His main area of interest is cyanobacteria, cyanophyceae members. But since he has associated with me, it has become a sort of contagious, and he started working on macrofungi. And then one day he brought coconut leaflet, and he showed me these things. And then I compared morphologically, and I told him that it may be a new species of Xylaria. And then further molecular studies and other things can be taken up by Madhav Kumar. He is scientist in Botanical Survey of India in Port Blair, and ultimately a new species has been erected in my name. It is very interesting because that is, I have already mentioned that the coastal region, scrub jungles are the hub of. So there are several that is the Xylaria species present, and it is very important to work on Xylaria, especially metabolites and the molecular studies and all such things. And this is with regard to mangroves. Uh, there are several species have been reported from mangroves, but one of the features with regard to mangroves is the size of the sporocarp will be small compared to scrub jungle or compared to Western Ghats, and even compared to uh, the coastal sand dunes. So you can see the Amaroderma here, and also Ganoderma is also here, and Hygrocybe, and Lentinus. This is edible, Lentinus paracellus, it seems. Lenzites are common. Lycopterodon is there, and then we have Picloporus, the highly pigmented species. Picloporus are there in the coastal triangle. This is the Lycoperdon species. So for our surprise, we got several species. And in fact, Acharya and his group from West Bengal has reported several species of macrofungi from the Sundarban region. And also, they have reported 2 species of termitomyces from Sundarban region. Therefore, so it is in our area, some of these species will be edible, and people are eating this, especially this lentinous species. What we got, we didn't get any termitomyces in mangroves. These are the common species found in the coastal sand dunes. As usual, the lentinus is very common, Lycoperdon, and also Pleurotus is here, Pleurotus. And also Termitomyces schimpferi, as I mentioned, Termitomyces schimpferi is very common in, and also Scleroderma and Theileria and such type of mushrooms are common because many of them are ectomycorrhizal, of course, in the coastal sand dunes, and interesting to work on various species. Not much of studies on the coastal sand dune and also mangrove macrofungi. There are several the hindrances with regard to climatic and human impacts on macrofungi, as everyone is aware. Soil erosion is one of the features. Floods are there, drought, and also many invasive plant species also responsible for human interference. That is, the invasive species will project because native species have been removed, or it has been because of thinning, or their representation will be less like that. And the second point, we have to look for the substrate removal. So a lot of people are removing the woody material from the coastal sand dunes, and that is detrimental for macrofungi. Not only this one, and they remove shell mining and sand mining. These are all some of the human interference that affects the macrofungi. Wildfire. Sometimes they put all this woody material in one place and lit the fire. So not only common fire, and even deliberately it will be done. These are all some of the human impacts on coastal sand dunes, and invariably it is going to affect the mycorrhizal and even that is macrofungi. The other reasons, what you can, tree felling, plantation activities, agricultural chemicals are used, and another niche abuse. This is very interesting. Niche abuse, nobody wants termite mounds. They remove the termite mounds because they are detrimental. It is not like that. So the termites are very, very important with regard to mineralization of lignocellulosic material, and also they are useful for us because they are going to give us termitomyces, edible and medicinally valued termitomyces. So that way, so niche abuse is there with regard to termite mounds. Expansion of exotic plantations, acacia, eucalyptus, and so some of this type of exotic plant, and even this silver oak and casuarina plantations are many, but invariably those will be cut after some time. So that is not going to perpetuate. So whatever plant species is going to be there and stay for a long time is very useful for macrofungi and other ecosystem development. Organic farming. In Germany, what I have seen, I have visited several national parks in Germany, and I see this, nobody can touch even a single leaf from the national park. But what they do, there will be a path where one can walk and see plants and animals or whatever there, birds and butterflies, something like that. So what they do, they simply heap this organic matter. Twigs, leaves, everything will be heaped in a different part of the pathway or inside the forest. That is, the heaping of organic matter is a lot of advantages because it is going to support a lot of insects, it is going to support a lot of animals, it is going to be cool, something like air conditioned like, and the animals can be happy with that type of heaping of organic matter. And we have not seen such type of heaping processes in India, but in some of the plantations in Western Ghats, I have seen coffee plantation, they are going to heap this leaf litter and twigs like that. And some people are aware that this is very important. Organic matter is very important. They are not going to remove the woody material fully. They are not going to remove the leaf material fully. Instead, they are going to use it as organic manure, and eco-friendly agriculture in the plantation activities are important. So I am going to project here with regard to fire. In case of scrub jungle, so this is the view of summer, peak summer, where lot of deciduous leaves will leave. Plants have shed the leaves, and it is almost barren like. And when the impact of fire, it is going to burn, and all this leaf material and even twigs and small twigs and all, it will burn. Some of the plants and shrubs are remaining here. But after rainy season, you can see lush green here established. With regard to the impact of fire, so if you look into, so this is entirely in the subjungle places. It's not a wildfire. It is a small fire. People are going to, sometimes they lit the fire like that. See, the species richness, if you look into the non-fire region, so it is starting with 2 weeks of wet season, and then fourth week it is more, and it is going to come down. So when fire attacks, what happens? The number is very less, maybe 5 species. Here it is 15 plus 5 species, and the peak will be shifted from 4 weeks to 6 weeks. 6 weeks you can see the peak and then come down, but not to the level where we can expect non-fire region. So with regard to sporocarps, see sporocarps, with regard to sporocarps peak is concerned, in the inception of rainy season, 2 weeks, 4 weeks, 6 weeks is the peak and come down. And here in the fire attacked, this is the non-fire attack region. 4 weeks it is going to be peak and come down. The fire attack region is going to be slow, and it is 2 weeks will be postponed with regard to their peak is concerned. But it is interestingly higher than the number of sporocarps what we see here. Species will be less, but number of sporocarps will be high in the fire attack region. So if you consider this, the non-fire, that is number of species found in leaf litter, 15%. Soil, it is 47%. Wood, 38% in the non-fire region. So all the leaf will be burned here. The fire region where we can find only two components, that is the soil, 54%, and wood, 46%. Some of them may be in the wood, and soil may be below the soil, especially mycorrhizal species may stay within the like. With regard to edible ones in non-fire region, 15% edible, 31% medicinal, 54% is ectomycorrhizal. So in the fire attack region, the edible ones will be increased up to 54%, and 31% in medicinal, and 15%, that is ectomycorrhizal, has decreased to 54 to 15%. These are the observations we made in the fire attacked region of the scrub jungle. So these are some of the fire tolerant mushrooms. So this is the Astraeus hygrometricus, and this is the young stage, and this is the mature stage of Astraeus odoratus. And Cyathus striatus is after fire, it is going to crop up like this. A Dacaryopinax and the wood material is going to grow, and then Lentinus parasolus will develop, and Palus merulinus and Paralus species will be there. And even you can see Termitomyces striatus also grow after the fire attack locations in the scrub jungle, very close to our campus. So now let us look into, I have only one slide now, richness and diversity. Species richness and species diversity has been plotted here, Western Ghats, and then 57 species, as usual, it is very high. Species diversity 3.06 to 4.58, because the species diversity depending upon number of species as well as frequency of occurrence. Therefore, it is very high here. West Coast 79, it is less than, diversity less. But coastal sand dunes have less number, maybe around 2.5 times less than Western Ghats. But the species diversity is very high, 3.6 to 4.16. It is almost higher or equivalent to that of Western Ghats. This is one, this aspect is bothering us very much, and we are trying to address this issue in the future. So why coastal sand dunes have so much of species diversity equivalent to that of Western Ghats, although number of species are less, depending upon the frequency of. And then mangroves, again, the west coast, that is the scrub jungle, and the mangroves are almost, number of species are high here, and the species diversity is more or less equal like that. And the core group species found 44 species have been reported from entire mangroves coastal region, and no much of overlap between the core group species. Core group species are very important because it is the one they are perpetuating very fast, and also their number is high because more than 10% frequency of occurrence, and they grow like. So the question is right: why Western Ghats and CSD have high diversity? So I am thankful to Mangalore University for sustaining me after my retirement too as adjunct professor. Then Myco India is very important to our heart, and Association of Fungal Biologists that is supporting us like anything. And these are the upcoming Myco India, Myco Asia, and the Fungal Biologists associations are coming up in a very big way, and to encourage a lot of youngsters and experienced people to share their ideas. That way, I'm thankful to Myco India and also AFD. My PhD students, Karan Chinnappa, is from Western Ghats proper. He is in the midst of Western Ghats. Therefore, we could carry out a lot of work on Western Ghats. Sudeep Ghate, from the coastal region, he has worked on the campus. He has worked, and Pavitra, another PhD student, worked on campus. And Grishma, also from Western Ghats, and she has carried out a lot of work on macrofungi. My MSc students, Dattaraj and Jagadish, have worked, and actually I was giving them lecture on entomopathogenic fungi and also cordyceps like that. And I asked them to look for the cordyceps near the somewhere where the insects are more like that. And one fine morning, Dattaraj and Jagdish came to me, and in the month of July, they about 4 or 5 years ago, they showed me cordyceps. Sir, these are the cordyceps we found in one of the places like that. Then we started working on that, and now it is almost 4 to 5 years of continuous work with regard to cordyceps. And we have one more person, that is Dr. Sharat Chandra, also involved in this. And every year in the month of July, August, we are hunting for the cordyceps, and we are looking for that. And thank you very much for your patience to listen my talk. And if you have any queries, you are welcome.
So thank you, sir. Thank you very much. What happened to my camera?
So we have to.
So thank you very much, sir. Can you please stop sharing, sir, please?
We have to do that.
I think you go to your slides. If you go to Google Chrome, sir.
I think it is full screen, you know, that's why it is. You cannot do it now.
No, so you go to Google Chrome, then you can not come back to Google Chrome, sir.
It is my system is not working like that. I don't know.
No. So it will work, sir. Agala, sir. Just go to Google, go to internet browser.
No, it is not showing anything because it is full screen.
Okay, escape.
Escape. Okay. Okay. Then
Google Chrome.
Okay, stop sharing. Okay.
Thank you. Still, it is.
Yeah.
Stop sharing, sir. That's.
Stop sharing. Yeah. Okay.
Yeah. Thank you, sir. Thank you so much for your wonderful presentation on macrofungi. What we interested here in this presentation was they talked about climate change and its possible effects on macrofungi, the diversity of macrofungi in the Western Ghats of India. I think I thought it could be a great. There is a great scope for research funding in this area. I believe. I am not sure. Um, we have a great baseline data about macrofungi fungi. I mean, we may have scattered information here and there, but there is no comprehensive systematic way of understand the diversity and the distribution of these macrofungi in the Western Ghats of India. So, what are your views about this, sir?
This is see, when we started our work in 2011, when we looked into literature, not much of quantitative information was not available. So if you want to talk about macrofungi occurrence, or if you want to compare Western Ghats with Himalayas, you need quantitative data. If you don't have quantitative data, it is very difficult. But quantitative data, Senthila Rasu has reported many agarics from Maharashtra, Karnataka, and Kerala. And others also, they have given quantitative information like that. Unless, so the species list and checklist is okay, no problem. One can do that, phylogeny, everything. But at some point of time, we need to give a quantitative information about the occurrence of a particular group of macrofungi. For example, we may talk about agarics, we may talk about cordyceps, we may talk about ectomycorrhizae, like that. Unless we have some quantitative information, it is not possible. That is the reason why I gave some information about quantitative information related to number of species and also their diversity and their the plant species preference. For example, ectomycorrhizae, as I mentioned, 143 species, and nearly 114 species are present in only Dipterocarpaceae group. That too, Veteria indica is very, very important plant. Therefore, so these are some of the information available, and people may take up the importance of Dipterocarpaceae, especially with regard to ectomycorrhizae is concerned. And in our campus also, we have several species of some 2-3 species of Dipterocarpaceae, but it is really wonderful to see some of the ectomycorrhizae associated with them, like Astreus hygrometricus, Astreus odoratus, and several species, even Scleroderma. They are all present in this one. One or two species are there, plants. Very few plants scattered here and there, but ectomycorrhizal species invariably you can find it, even in the fire attack region also, because the tree will stay, and next crop will come with the ectomycorrhizal species will crop up.
So, I mean, after working on this group of fungi, groups of fungi, do you have some groups or some taxa in your mind that can be used as a keystone species or a reference taxa with respect to climate change and its possible effects on macrofungal diversity of the Western Ghats of India?
Yeah, that is. So there are in the list there are some sensitive species are there. Some species are very sensitive, and see that as I mentioned, Western Ghats and West Coast and other regions, even in the Western Ghats, sacred groves, reserve forest, and the Shola forest. There is no overlapping like that. Each ecosystem is independent, and I'm sure there will be some sensitive species pertaining to tree felling and road construction and mining, granite mining and such type of things are happening. Railways, these are all. So by that, some species may be lost because of such type of activities. Especially tree felling is very dangerous because a lot of ectomycorrhizal species may be lost because they may not have plant preference, but if you lose some plant, very few plants are there. Even you know very well about the mangroves. The mangroves specific plant species are deteriorating too much, and we may count some species in fingertips. So if you lose it, automatically the associated fungi will also be lost like that. And even Western Ghats also may have the same story.
Thank you, sir. Thank you very much for those insightful, responses. I feel, there is a great potential, great scope for research funding in this area, because there appears to be lack of sufficient baseline information, baseline data about the diversity of not only the baseline data, even the quantitative data seems to be very limited. So, but to make a meaningful conclusion, or to make a meaningful statement about the possible effects of climate change on this macrofungal diversity, we need to do more, and we need to do very smartly, I believe. With this, I request Professor Bhatt to ask any questions if he has. Professor Bhatt?
Thank you, Damodar. I have Professor Sridhar. It's a really nice talk that you gave, and very nice. Very happy to have heard this. Just for curious, some time ago, just a couple of months back, this made a news even in the international newspapers about one mushroom sitting on a frog.
Frog, yeah.
In Kerala. What, it may be, accidental, or it may be a general feature.
Huh.
But so much was written about this.
So it is curious information, and I have also seen that picture very nicely. That mushroom has grown from the upper surface of a frog.
Frog, yes. But it's in Kerala, and our Dr. Pradeep gave some idea about that, why it is happening and all. And I read that about even in some international newspapers, as a issue. That means what it gives us an idea is that, there is so much of curiosity about fungi. And we have not taken this to, I'm telling about last 4 or 5 decades, we have been working on these fungi. But the awareness it should come in the younger age. We should take this to, instead of working like a class apart, we should go to, I'm telling this to Damodar also, we should take this our children, to if some curiosity, awareness, and the importance of fungi goes to that level, I think conservation will come automatically. Otherwise, what people know or speak about this only plants and higher animals.
Yeah.
Just a wide thinking, which I thought I'll put across. But again, as Damodar has put little earlier, disappointing. The number of people who are participate today is truly less.
Very less. 34 now.
Yeah.
Anyway, Dr. But it is very good that you have commented. With regard to cordyceps, what we have found about 5 years ago, 4 or 5 years ago, that is from the Arboretum or the Vijaya Bank Botanical Garden.
Yes.
That's been raised by Dr. Chandrasekhar. I should be thankful to Dr. Chandrasekhar. Whenever I go there, I can find one or the other macrofungi. And even in the month of July, if I go, definitely I will get 1 or 2 cordyceps, known or unknown species like that. So therefore, that shows the importance of this arboretum and botanical gardens.
Correct.
You know very well that Professor Kaveriappa is responsible for developing the arboretum.
Yes.
Several endemic and endangered plant species are there. And if you look into the seasonal variations with regard to the macrofungal this thing, arboretum will be an outlier. It is something which is higher than what we can see in the botanical garden and scrub jungles. But that probably that type of ecosystem is very, very important. And also the plant species, what they have housed in arboretum, also may have major role in that. In that context, I am thankful to Professor Kaveriappa as well as Professor Chandrasekhar. I think you were also there at that time when Arboretum was.
Yes. When it was established, I was there briefly. I was there in Bangalore. Yes. Yeah, it's a wonderful thing. Thanks. Yes. Thank you.
Thank you, Professor Bhatt.
Thank you, Professor Bhatt, for that question and comments. About the disappointment about the number of participants, I would like to highlight here that the 43 people who joined in today, they are the genuine knowledge seekers. And we are very much happy and these 43 people are here, and they are enjoying this wonderful lecture and the discussion here. Of course, as in Myco-Asia and Myco-India, I mean, quality does matter, not the quantity. Of course.
So, yeah, you are always telling that.
Dr. Sridhar, Dr. Ananda wanted to. First of all, he complimented you, saying it was a wonderful presentation.
Yeah,
He wanted to know, can you give some reasons for increased species of edible mushrooms after fire?
After fire, that is, I'm not very sure. That has to be worked out. But see, what happens, our campus, you are in that campus, and every summer there will be some fire. It may be deliberate or non-deliberate. But it is a sort of curiosity for me and my student. And why can't you work on this fire-impacted region line? She selected 3 fire-impacted region of scrub jungle and worked like that. And probably the ectomycorrhizae also increases because they are protected very well in the root system. Although surface fire will be there. And with regard to edible mushrooms, I don't know, because we have classified based on edible, medicinal, and ectomycorrhizae like that. We have given a sort of pie diagram.
Is it a one-time study, sir? Is it a one-time study?
No. It is. We have carried out in triplicate and actually one-time study. In fact, one-time study.
Okay.
No work has been carried out like that. So even Western Ghats also we can take up such type of work because invariably you can find fire in different part of Western Ghats. And nobody is bothered about looking into that like that. The only thing is if you have a baseline data, then we can go to that region and see what happened after fire.
Or these fungal spores have fire resistance capacity?
Yeah, maybe it is not according to me, it is not the resistance of spores. It is instead, it may be hiding in the woody material or it may be hiding in the root material. Like that, then it may crop up when the conditions are favorable. It is going to crop up. It may be sort of endophytic also sometimes.
Thank you, Dr. Sridhar. When I was doing PhD in Hong Kong, for a couple of months I was wondering whether I should be studying about the effects of fire on fungal diversity. I actually did some literature survey. It looks like after the fire incident, the organic matter increases because of the death of some plant species. So increased amount of organic material available for the fungi, that might also probably results in the increased biodiversity of fungi. That could be another reason. And there would be less competition from the plants for the same amount of or not for this fair amount of the nutrients there, I think.
And even phosphorus content will be high in the ash. Therefore, that is another important thing.
I think Dr. Anand asked a very good question. I think it's very thought-provoking, and Dr. Sridhar, as usual, gave a very interesting and insightful response.
Even in farms also, they fire it and they lit even sugarcane. They burn it and then they grow for the new crop. Like by that, the phosphorus content will be high. The productivity will be high unless and otherwise there is no erosion. So unfortunately what happens in our terrain in the west coast, there will be torrential rain and all this, whatever ash will be taken away to streams or estuaries like that. That will be enriched rather than enriching this one, unless we have some breaks of this thing and some obstacles to remove the ashes and such things.
Okay, ladies and gentlemen, we will take up 3 questions from the three audience, 3 people from the audience. First question will be from Dr. Seshireka. Dr. Seshireka, please ask your question.
Yeah, good afternoon, sir. It was a wonderful talk. It has also created more interest in this group of fungi. I just wanted to tell you one this thing. Ophiocordyceps type of thing which you had shown in the slide, the orange colored one, found one in the Sanjay Gandhi National Park. At that time, I didn't know it was Ophiocordyceps or what it was. I thought it was a small mushroom, and I collected because it was raining heavily, and we couldn't find the insect or whatever it was growing on. It was too wet. It just came off. So that is one thing. Is I don't know if it is reported from the Sanjay Gandhi National Park here in Maharashtra. That's one thing I wanted to ask. Then there was one more thing which was already eating my brain. Like, I had read one article about this Schizophyllum commune, which grew from a lady's hand, the fruit bodies which grew from lady's hand. Like, many times my students ask whether it is infectious if we handle any mushroom, and if it will go into our skin and it will start germinating and all. How possible is this? I know it is not so this thing. Our bodies are not so irresistant, but then is it possible? It's that way.
That you are talking about the cordyceps, no? Cordyceps.
Cordyceps was one thing. I have not confirmed that yet, but it looked similar from your image. And my main question was this.
See, this especially the cordyceps and related species are attacking the insects, and many occasions what our experience says that the insect will be that they are burrowing the caterpillars, and even some of the adult insects also below the soil sometimes, or lot of leaf litter is falling on the surface, and when the conditions are favorable, like heavy rains and humidity and temperature are congenial, they start cropping up. And while taking such type of fungi, we should be very careful, and we have to dig it very nice.
I wasn't know. I thought it was a nice tiny mushroom.
We cannot simply take from the surface like. So that we have to dig it. At least we have to go for a few centimeters below. By that we'll get the. That's how we got the caterpillar and the insects and testing.
Yeah, we are hoping this season we'll see it again, and we'll collect it the way you said. And what about this Schizophyllum, sir? Is it true? Is it true? I mean,
Schizophyllum that grows especially on wood, and also it is an endophytic fungus.
Yeah, but there was an article which grew from a woman's hand. I mean, it was growing from a human being.
Yeah, that's also I have seen. Almost like the frog.
It was growing so much, and it was really scary.
So I'm sorry. I have to stop this discussion here. Let us move on to the next question now, because we have got only 7 more minutes, Dr. Sridhar.
It's okay.
Dr. Sunil Deshmukh is just watching me. Tisha, please ask you.
Yeah. Hello, sir. Yeah, it was very nice topic and a nice session regarding cordyceps. And moreover, I'm working about cordyceps militaris and as well as cordyceps senesis. Just I wanted to know about how our cordyceps will help in future generation with respect to diabetic patients. Means when I'm gone through for cordyceps articles, cordyceps is very helpful for reducing type 2 diabetic patients.
Yeah, there are a lot of literature is there on the impact of cordyceps on diabetic patients. Probably Deshmukh may be in a position to will give some highlight on this.
Actually, polysaccharides present in the cordyceps, they are responsible for reducing the diabetes, particularly in diabetes type 2. But there are not much literature on type 1.
Yes, sir. Yes, a lot of articles having type 2 diabetes with respect to cordyceps. Yeah, since I am working out, I'm making some formulations for cordyceps kind of things. Means cordyceps tablets kind of things, to working out how cordyceps will reduce type 2 diabetics for the patients.
But we have to check whether any toxicity is related with that, because most of the experiments are in vitro and very short term. We have to check long term, about a year or so, any side effects are there or not.
Yeah, okay.
In fact, cordyceps in China, it is and Southeast Asia, it is taken directly along with the food like that. It is more of consortium rather than isolation of a specific metabolite. And isolation is very, very important with regard to pinpointing what type of metabolite is going to control the human illness like that.
Yeah, okay.
Thank you. The last question from Dr. Devdatta, please.
Good afternoon, sir. Very nice presentation, sir. You have covered from terrestrial to marine environment. So I have a question regarding that marine environment, especially have you reported the diversity from the aerial parts of the mangroves? Because in the intertidal environment, we found very less diversity, Halosipina or Nia vibricia and other species. So what is your comment on that?
Yeah, that is, see, actually, we have got, recently we published a paper on the canopy, that is the mangrove canopy. There are some marine fungi present in the canopy. We got the, we collected the through fall and also stem flow from, I think 2 or 3 plant species, Vicinia officinalis and also Rhizophora mucronata, and also from the tree holes like that. And we got some typical marine fungi. We got that is the Nia vibrisa, Nia species, and also we got a few Perlospora species like that. And that is, I think it is a sort of intermittent habitat where they are interacting. They are all involved in decomposition like that. It is interesting to know how this marine fungi present in a terrestrial situation, not completely terrestrial, at least partially terrestrial situation. This marine fungi also exist.
Yes, sir. Actually, I used to find a lot of times Ganoderma and some Auricularia species are quite common. Whether I still believe like whether we can report them as marine or else like facultative. I mean, that is very quite confusing for me. That's why.
So you need not necessarily to report it as marine. Where you find it, we got it in mangrove habitat. And if you give some information about the salinity, season, and time of collection and such things, that is more than enough, because it has some salinity effect if it is in the summer season. If it is in the rainy season, it may not have that much of salinity. So salinity, pH, and temperature, and even other features also can be reported. The water quality like that, or the soil qualities also can be reported. By that, you need not worry whether it is marine or terrestrial. You need not necessarily to decide about it. You got it in that ecosystem at that point of time. Okay. That is going to pave way for others to look for other reasons. Why they are growing in mangrove, why they are growing in coastal sand dunes, why not here like that?
So one more question is like we have reported from few diversity of cordyceps, especially Opiocordyceps nutans and other species. So whether you have evaluated that medicinal properties or bioactive potential of them and by culturing them?
No, I have not cultured them. We are not cultured, but we are working on that. And our experience is every year we go for that. We are getting new type of cordyceps. We don't know whether it is nutans or Cordyceps sinensis like that. We have to go for molecular studies only to solve the problem. Right. Some of them we have identified based on the morphology, but molecular studies is very much important.
Thank you, sir.
I think thank you, Devdatta, for those questions. I mean, about Devdatta's, pertinent question is whether how do you differentiate marine fungi from the boundary is becoming a little bit not what you call foggy, and probably we need to fine tune our terminologies, marine fungi, terrestrial fungi. Maybe we could say fungi from terrestrial environment, fungi from marine environment, fungi from mangrove environment. So when you say marine, that means the marine systems are owning it, not like that actually. Anyway, thank you for those thought-provoking questions. The final comments from Dr. Sunil Kumar Deshmukh, if any?
Actually, this was very nice presentation. I was expecting more people. If you talk of cordyceps, particularly in the Mumbai till Konkan area, we get around 8 to 10 species.
Yeah.
But we have not published so far. And secondly, about Schizophyllum, if we talk of Schizophyllum communis, we have got 2 cases where they got the infection on the skin, but mostly this is because the farmer got the injury. And because of injury, they got some these were having diabetic patients, so they got the infection. This may be one of the reasons, that one is injury and second one is immunocompromised person, so may get infection. Normally, healthy patient, we have not heard anything. So it was nice presentation, and thankful to him for making so nice presentation.
Okay, thank you. Thank
Thank
you.
you. Thank you. Thank you very much, Dr. Deshmukh, for those final comments. I would like to take this opportunity to thank Dr. Kandikere Ramaiah Sridhar, the editor-in-chief of Myco India, Journal of Indian Fungi, for this wonderful presentation. Also, very interesting discussions we have had. The number of participants may be disappointing, but definitely the discussion has been top class, as we always expect us to do a wonderful discussion with our participants. And I would like to thank the Association of Fungal Biologists, Dr. Sunil Kumar Deshmukh, Dr. Shashi Rekha, Dr. Mahavir Gosavi, and some more wonderful people there, and under Professor Bhatt and some other eminent scientists participating here, and all the ladies and gentlemen in the audience for your kind participation today. As I mentioned,
I want to thank Dr. Isabel. She is from Portugal. She has attended this talk. She is one among the audience. Dr. Isabel Fernandez from Portugal.
Yeah, thank you. Thank you. And as I was mentioning, you are the real knowledge seekers, 43 people or 45 people who participated. That too on a Saturday. Normally our programs are on Sunday, so we normally get up 50 plus people. As it is a Saturday, and there are not much attraction like participation certificates or Amazon vouchers. Still, you are participating today with the pure intention of gaining some knowledge. You are very much, your presence and your participation is very much appreciated. With this, we sign off. Thank you very much, ladies and gentlemen.
Thank you.
Thank you.
Thank you. Bye.
Bye.
Bye, Deshmukh.